and welcome to Science Project 101. My name is Rachel Peterson.
Hello, I'm Emma Cushman.
And I'm Amy West.
In this video, we are going to explain the Science Fair.
The backbone for all science experiments is the scientific method.
The first step of the scientific method is state your problem.
Basically, just state the problem is a question that you would like to find out and do your experiment on.
Make sure you pick something that interests you and that excites you.
The next step is
Gather information. Gather information is researching all the information you're going to need for your experiment and to write your report.
Don't worry, we'll explain the report later on.
The next step is formulate a hypothesis.
Your hypothesis is an educated guess of what you think will happen in the experiment.
For example, if your experiment was, do snails prefer smooth surfaces, then your hypothesis could be yes or no, depending on what you think will happen. Make sure to put why you think that will happen based on your research.
Don't worry if it turns out wrong because this is all just a learning experience.
The next step is
Test your hypothesis.
This is when you actually perform the experiment. Let's use the one we were discussing earlier. Do snails prefer smooth surfaces?
This is when you would actually place 3 snails on a smooth surface, such as a smooth block of wood, and 3 snails on a rough surface, such as a rough block of wood, and see whether they actually do prefer the smooth surface.
To make sure your results are correct, do the experiment several times.
Remember, you do not have to do the experiment we just suggested. Be creative. Originality is the key to a good experiment.
We're almost done.
Record and analyze. A scientific journal is a place where you keep all your data and observations. You need a scientific way to measure that's not by opinion, but by scientific observations. Make sure you record your data throughout your whole experiment.
Last but not least,
state the conclusion. In your conclusion, write whether your hypothesis was right or wrong. You should have data to back up your conclusion, state any mistakes you made, and explain what you would have done differently.
While you are performing your experiment, be sure to maintain the following: constants, variables, control, and experimental groups.
Your experimental group is a group you actually perform a test on, while the control group is a group that is used as standard for comparison.
And constants are the conditions present in both the experimental and the control group, while variables are only present in the experimental group. Now we will explain how to present your work. You already have the title, conclusion, and hypothesis.
Your results, it shows what happened in your experiment. Make sure you have a graph which shows how the thing you're measuring changed or did not change.
Your procedure is exactly what you did in your experiment. Like step 1, I gathered all my materials. Step 2, etc., etc., you get the point.
And materials are just all the things you used during your experiment.
A Bible connection is a memory verse from the Bible that relates to your experiment.
Your purpose is why you're doing the experiment. It's better to have some data to back up your purpose.
And the report is a 300, 400, 500 word essay, depending on your grade, on an element of your experiment.
Last but not least, we have the resources, or otherwise known as the bibliography, which is a list of all the places you got your information for your report.
When you're presenting your board to the judges, you're going to want to state your title, and then your purpose, why you did the experiment, and then go to your report and say the research that you did and things that you learned during your experiment. And that will lead you to your hypothesis. Say your hypothesis and explain your hypothesis, Then your procedure is what you did. You just say your procedure. And then you go to the result, what happened to your experiment. And I chose a bar graph for this because this is clearer than some other graphs you could use. And then your conclusion, just say what Rachel said, like whether your hypothesis was right or wrong and what you would have done differently. And then Bible connection, state your Bible connection and then say why you picked it and how it relates to your board.
Now I present my board to you as if you were the judges at home. Hi, my name is Rachel Peterson, and my science fair project is building blocks for healthy bean plants. What type of fertilizer causes the best overall growth in bean plants? The purpose of this experiment was not only to see which type of fertilizer causes the healthiest growth. I had Alaska, Schultz, and Miracle-Gro, but also to see if the amount of nutrients, which are key to a plant's growth and health, had any visible effect on the plant. In my report, I learned a lot. I learned that nutrients are divided into two groups, macronutrients and micronutrients. Macronutrients are the nutrients that the plants need more of, while the micro are the nutrients that the plants need less of. I also learned that the amount of nutrients a plant gets can really affect the plant. For example, if the plant does not have enough nutrients, it will start dying and withering. If it has the correct amount of nutrients, it will be a healthy green color. But if it has too much nutrients, the plant will grow too fast for the roots, and it will probably tip over and die. I also learned how plants actually receive their nutrients. They receive their nutrients and water through their vascular systems, which are in the roots. The little nutrients climbs up through the root hair into the stem through the xylem tissue. Capillary action helps the nutrients get up the xylem tissue. Some scientists also believe that when the plant needs the nutrients and the water, it actually pulls it into themselves. I also learned that plants not only need nutrients and water, but they also need food created from photosynthesis. It starts when the sun shines on a leaf. On the leaf, inside is a little cell called chloroplasts. Chloroplast stores the sunlight and makes chlorophyll, which then makes a chemical called ATP. Eventually, through this process, ATP becomes glucose, which is what the plant needs to survive and for its food. So how does ATP come and become glucose? Well, it starts out when water comes through the stem and into the leaves. Then the water breaks down into hydrogen and oxygen. After that, the air brings in carbon dioxide. And the carbon dioxide combines with the hydrogen to make glucose. So that was just a tiny bit of what I learned in my report. My hypothesis was that the Schultz plant's food would help the bean plants the best because it had a ratio of 10-15-10, and that means it has 10% nitrogen, 15% phosphorus, and 10% potassium, and those are all very key macronutrients for the plants. While the other plants, like the Miracle-Gro, had a ratio of 8-7-6, and the Alaska had a ratio of 5-1-1. My procedures was first I bought 12 bean plants. Then I bought the Miracle-Gro, the Alaska, and the Schultz plant food. After that, I transplanted each plant into the same size pot with the exact same amount of soil and kind of soil. After that, I labeled each pot with their name and their plant number so I could identify them when I was doing my recording. After that, I mixed water with each fertilizer So water was the constant. All the plants were fed water, but just some were added in different types of fertilizers. After that, I fed each plant with 1/3 cups of their designated fertilizer or water a couple of days every day, and then I rotated them because they were put in front of a large window full of sunlight, and they're in rows so that they could get even amount of sunlight. After that, I recorded my data in my science journal. Here, I measured a couple things. I measured the height of the plant, the width of the largest leaf, the number of large, small leaves and buds. This is my first recording. And then I did all the other days, but not with pictures, but in tables. Now I come to my results. These are the overall percentages of the growth from the beginning to the start for the plants. Water had 106% growth. Alaska had 85.9% growth. Schultz had 148.5% growth. And Miracle-Gro had 163.9% growth. From this, you might think that the Miracle-Gro, which had 163.9% growth, was the best. But I saw differently, because 2 out of the 3 Miracle-Gro died, and so that would be only one variable. But while the Schultz, all 3 lived and all did well. Here are some of my graphs. The main one would be this one. This is basically all the heights. You can see how fast they grew from Miracle-Gro and Schultz. But I did not include the dead. I did include the dead plants in my graphs, but not in my percentages. So my overall conclusion was that the Schultz plant food did work the best, because even though the Miracle-Gro had a better and higher percentage, at the end, the last Miracle-Gro started dying and drooping, while the Schultz did amazingly. My Bible connection is, let my teaching fall like rain, and my words descend like dew, like showers on new grass, like abundant rain on tender plants. And that really spoke to me and connected with this experiment, because God was relating his mightiness with a little plant that might not seem very mighty, but it's so complex, and he's just amazing for creating those.
At the end, we will show you some websites that will hopefully give you some ideas for your scientific science fair project.
Hopefully, we gave you some idea of what the science fair is and how to present and perform your experiment.
Thanks for watching!